Great, thank you. Yeah, let me know if there's any problems with seeing or hearing. But thank you so much. I'm excited to be here speaking, even if virtually, about my recent work trying to quantify the extent to which we can trust database annotations of metaomic sequences for microbial eukaryotes. So as we've heard a lot about this week, and especially in the last couple of days at this symposium, microbial eukaryotes are particularly underrepresented in databases, which can pose a lot of challenges to annotation. And this is in part because protists have diverse genomes that have many unique coding and non-coding sequences, as well as the fact that protists are comparatively understudied with relatively few isolates of protists having been sequenced, in particular genome sequencing efforts as compared to, for example, transcriptome sequencing and collation efforts. And because of this, it's standard and expected for, in many cases, upwards of 50% of sequences in metaomic data sets for microbial eukaryotes to go unannotated, which is a large problem, a challenge for accurate interpretation of these metaomic data sets that we're using to try to answer environmental questions. So to illustrate this with a bit of an example, database composition directly determines the annotated labels that we get out of our transcriptome or a metatranscriptome or metagenome annotation efforts. So in this example, we have a database that contains different types of fruit, an apple, an orange, and a banana. And applying this database to a set of query fruits, in this case, a guava, a peach, and an apple, we might get different labels depending on our database composition. So starting with this first database, this guava, we might say, looks reasonably like an apple, has a similar shape, maybe it doesn't match in color. The peach we might call an orange, and the yellow apple, we might not be sure. If we prioritize color equally to shape, we might not be able to tell the difference between a banana and a red apple, since it matches the yellow color and the apple shape. If we add a new entry to our database, in this case, a yellow-green guava, our annotations might change where we have some ambiguity in different groups. So now our green guava might either be an apple or it might be a guava of a different color. And the yellow apple now looks most similar to that guava. So depending on what we know as true fruits, we might annotate the fruits that were given as query fruits differently, the same way our query sequences might be interpreted differently on the basis of what sequences that we have in our database that we've seen before. And one major advantage to studying microbial eukaryotes in comparison to the challenges with studying them related to database completeness is that we can identify eukaryotes by eye. They're visible in many cases. So this is an opportunity to be able to compare metaomic sequences to microscopy to evaluate how well we're doing at taxonomic annotation in these data sets. In this example set of sequences or set of samples where we did sequencing as well as microscopy, we had 4 different sampling dates in Narragansett Bay, which are the different columns, different bars on this plot, where we had microscopic counts conducted. The bars here are relative abundance on the left y-axis, and the points and lines are cells per liter absolute counts for these different sampling dates. And you can see that there was one sampling date where we had a big bloom of one diatom family in the red here. This is Skeletonium metaceae. And on the other sampling dates, we sometimes had high concentrations of these couple of other diatom families, Rhizos lineaceae in yellow and Hemielaceae in orange. And one caveat here is that we did do metatranscriptomics. So this is community expression, looking at RNA rather than looking at community composition directly. So there may be some fluctuation, but in general, we can compare the trends that we see in the breakdown of the community in microscopy to what we get in these community level sequencing data. And what we see is that while we recapitulate our pattern of this large Skeletonium metaceae bloom, we have a lot of other sequences in the gray that are other taxa that we didn't record in our microscopic counts. And we also have this discrepancy of this Rhizus lineaceae family in the yellow, where we don't really observe it at all in the meta-omic data as compared to it being a dominant member of the community in the microscopic data. And again, while we expect some fluctuations between community level expression and the true community composition of individuals, we would expect that we at least get a fair chunk of sequences in this family Rhizus lineaceae, given the abundance of individuals that were identified in the microscopic counts throughout the time series. So we might look at our database composition to try to get some answers about these discrepancies that we're seeing between taxa. First off, these black bars tell us how many representatives we had present in the database for each of the major diatom families that are listed here. And this very top bar is Rhizus lineaceae as compared to this bar down here being Hemialaceae. And what we see right away is that we don't have few representatives for Rhizus lineaceae compared to Hemialaceae. We actually have 3 times as many, and we have an exact match to the organism that's present in Narragansett Bay for family Rhizus lineaceae, as compared to having a relatively distant relative for Hemialaceae. And within Hemialaceae, we actually have a lower comparative average percentage identity between sequences in the same family for that group as compared to Rhizus lineaceae. So nothing in particular about our database composition is explaining why we had this big discrepancy in not being able to identify a major group of diatoms. And looking at a slightly different example of an environmental transect where we had a fair representation of these radiolarians, so a group of microbial eukaryotes, we wanted to evaluate something of that example that I showed earlier where we added in that guava of whether or not adding radiolarians to the database when we originally conducted the analysis using a standard database that does not contain radiolarians changes our interpretation of community composition along this transect. And we expect for community composition to vary when adding radiolarians in one relatively straightforward way, which is that sequences that were previously unknown that are radiolarian sequences should now come up as being radiolarians. But we want to see whether there are other change-ups to the community. And so we evaluated this by annotating the data set first using a database containing just sequences from the MAREF database and the Marine Microbial Eukaryote Transcriptome Sequencing Project database, and then adding radiolarians. So the black bar here are those unknown sequences. So the scenario that I just described, where the sequence was previously unannotated, and then after adding radiolarians to the database, the annotation of these sequences changed. And you can see that about half of the total number of radiolarian sequences here labeled in blue belong to this category that were previously unannotated. But the other half of all these sequences came from other taxa, either one of these major groups that I've labeled in the warm colors, or some relatively low representation groups labeled in the gray. So a fair number of sequences, about half of all radiolarian sequences, were changes. So things that we previously thought that we could confidently annotate before radiolarians were added in, but now we've changed our mind and we say they might be radiolarian sequences instead. And a fair number of these other sequences also end up switching to the unannotated category. So because of a conflict, something that looks just as close to a radiolarian sequence as to some other taxonomic group, we've now actually reduced our confidence in annotating a proportion of the sequences in the data set, changing our interpretation of the community structure. And we might think this only applies to these higher trophic levels, so coarser taxonomic groups, but this actually is also a problem at the genus and species level when we have representatives in our database from a given genus, but maybe different representatives from within that genus at the species level. So I tested this using the genus Phaeocystis, which lives in either colonial forms or free-living organisms. And there's a division between species of Phaeocystis with respect to whether we often observe them living in this colonial form. Phaeocystis antarctica, Pouchetti, and globosa tend to live as live in colonies, whereas Johnny, Chordata, and Rex tend to only live in that free-living form. And I use databases containing either just free-living Phaeocystis, just colony-forming Phaeocystis, or a combination of all of the references that I had in order to annotate data sets or the Tara Oceans data set, specifically from the Southern Ocean and from the Mediterranean Sea. In the Southern Ocean, our dominant species of Phaeocystis is Phaeocystis antarctica, which is one of those colony formers. And as compared to using a database containing all the Phaeocystis, we recover most of those sequences as being genus Phaeocystis when we use the colony-forming database. But we do a very poor job when we only have Phaeocystis species in our database that are free livers. So if we only had a free-living Phaeocystis species in our database, we would have missed a lot of the sequences that we are positing are Phaeocystis sequences when we have the right species in our database. Conversely, in the Mediterranean Sea, where Phaeocystis cordata is the dominant species of Phaeocystis, we have the opposite scenario. So database composition can have a really strong impact on our ability to annotate and interpret our samples. And I won't have time to really talk about this in detail today, but I have worked on a solution or a path forward towards annotating sequences with higher confidence, or being able to interpret uncertainty in taxonomic annotation that I call taxaliquots, which is a method of using clustering rather than alignment to constrain the uncertainty in taxonomic annotation of sequences in meta-omic data. And this, as well as the 3 vignettes that I've talked about so far today, have all been recently published in a preprint on bioRxiv. And this actually expands the total number of sequences that we're able to annotate as some taxonomic group. So it reduces the overall size of the unknown fraction of sequences, both using this Tara Oceans example, as well as using those environmental data containing high frequencies of diatoms. So with that, I'd like to thank all of my collaborators on this project, in particular, Dr. Frederick Schulz and the New Lineages of Life group at the JGI, who I've been working with for a couple of years now, which was enabled by a couple of practicum experiences at the JGI through my graduate fellowship, as well as my co-advisors, Harriet Alexander and Mick Follows. And I'd be happy to take any questions I have time for.
All right, so we have time for one question, maybe? Okay, I'll ask you a question then. So Arianna, for the Phaeocystis issue, was that, just be clear, was that a database composition issue, or are you just saying that they sort of switch at random when you're sampling different data sets?
Yeah, so it was that for the database composition makes a big or has a big influence on whether or not we're able to accurately recover Phaeocystis in general at the genus level. So whether or not we say Phaeocystis is present in the data set hinges strongly on whether or not we picked the right Phaeocystis species to add to the database. So basically this is saying if I randomly picked, I said I was just going to include one genus Phaeocystis species representative in my database to reduce the effort of annotating my sequences or something of that nature, then depending on which one I picked, I may or may not accurately annotate Phaeocystis in the data set. And that's because of this division between the more genetically similar colony formers and the more genetically similar free-living species of Phaeocystis.
All right. Well, thank you very much.
Yeah, thank you.